he was not able to make it. And he sent me his slides, in fact, and we can put them online, but there's no way that I can really do his slides justice. I mean, his breadth of knowledge on psychiatric genetics is just impressive, and it would be dreadfully boring for me to sit here and read his slides. So what I did instead was actually do basically a quick summary, and this will be much shorter. We'll have time to continue chatting before the shuttle arrives, or we probably even have enough cars here to get everyone back to the hotel if we need to do that. And just instead do some quick, summary of what you saw today. Can everyone hear me? Yeah, good. And there's a clicker. All right, so these are the 4 talks that we had. We had Vincent talking, starting with that with an excellent review of the foundations of how all this is based on, and that the theory that underlies SOLAR. And then we had Peter giving an overview of solar eclipse and the project and its sort of its reason, the reason d'etre, which is extending SOLAR to imaging phenotypes. And then we had a great talk from Neda on the meta-analysis and mega-analysis, and then we concluded with Bennett's basically, really showing you that there is something here. This is not vaporware. We've actually been doing some work. So I'm just going to give you a couple slides from each one of those talks, just kind of the highlight, and in this case, edited. And so basically, just if I have to say that anytime you mention multivariate statistics, I think for a lot of people, their eyes will glaze over, and it's difficult stuff. But I have to say these things are actually really, really crucial, and they are fundamental to understanding what's going on in any sort of heritability analysis. And so basically, what SOLAR is doing at every single phenotype, that for every single voxel, every single region of interest, it's fitting a linear model, which hopefully we're okay with. This is like age, gender, what have you. These are the nuisance effects. And the key thing is what it's doing with what's left over, which is a genetic effect and an error that's of non-genetic sources. Now we can't individually get at these. It's kind of weird to write this. We couldn't come up with an estimate of what is the genetic effect for one individual. What is kind of the genetic residual? We cannot get at that. But what we can get at is the covariance that these terms induce. And so this is just the most essential takeaway from Vincent's talk, which is that we decompose the variance of the phenotype into these 2 terms, which is this part is the kinship matrix. So this is basically saying how similar are different individuals. And this part is just an identity, and this is the residual environmental variance, the variance that's unique to each individual. So again, that's just a picture, but the next slide, I have an expansion of what this looks like. The key thing is this would not work if we had independent individuals. We are depending on the fact that we have related individuals. And here is a slightly more complicated pedigree that I had a slide of this sitting around. Showing you what this looks like. And of course, if we represent everyone, we'll start with the founders. So the founders are the people that are not related to anyone else, and so they're shown here. So there's 0 on the kinship matrix, and these founders then gave rise to some families. And in particular, these first 2 people had 4 offsprings, as you see here, because they all have 50% genetic relationship. So it's this weird thing, because we have pairs of chromosomes, officially the kinship matrix phi is sort of what's going on for this. So the average genetic content on one chromosome. So we double it to get what the information on the pair of chromosomes. So it's just, usually we just write this as 2 phi. And you can just think about what is the genetic relationship. Well, how much genetic material on average is shared between each pair of individuals? And this is saying between this parent and this individual, that's 50%. So this is, going back, this is key. So we're basically saying that if we have a highly heritable trait, if there's a lot of genetic variance that can be explained, we're going to be looking for not independence, we're going to be looking for dependence. We're going to be looking for correlation. But it's not just any correlation, it's a correlation that's going to exactly follow this pattern. And that's the key thing. And so this is not any pattern, it's a very special pattern, exactly induced by the relationships between the individuals, and this is just absolutely essential. And of course, that's why it's important to just get all the details right in terms of specifying the individuals and specifying their relationships. So anyway, this is the fundamental to everything, and this is the magic that Solar is doing, is estimating these two parameters. How much variability is explained due to sources of variance that do co-vary with a relationship, and sources of variances that do not. Okay, so take home, just in words here, this is what I just said. We cannot measure heritability unless we have varying degrees of relatedness. And that is key for estimating this sigma g parameter, and then it's, of course, this ratio, the variance of the genetic variance to the total phenotypic variance, which gives us heritability. The other thing to note is that you might say, well, you know what, I'm not actually that interested in heritability. I'm in the Human Connectome project, and that's a neat project where we have families of 4 or so individuals, and there's always at least one twin in each family. But the vast majority of the Human Connectome investigators actually don't care about heritability. They just want to analyze their high-dimensional connectome data or the DTI data, and for them, heritability is just a nuisance. Well, this is indeed a nuisance, because if you ignore the heritability and you're working with something that is heritable, you're going to get correlation. That's what is driving the estimate of heritability. And so that will bias your standard errors if you ignore it. So SOLAR is important not only if you want to estimate heritability, but if you want to get safe, unbiased estimates of effects like age, gender, education, whatever, to account for that. So that's another important role for SOLAR that maybe wasn't amplified. But if you are stuck with data of related individuals instead of sort of embracing it, you still need SOLAR to give valid inferences on the covariates or group differences. From the next talk, this is from Peter's slides, and this is a great example of we think about fMRI, we think about MRI as a quantitative measure, but I mean, can it really be the case that the FA in this population of individuals is 2 units, 0.2 units different from this other population between the GOBs and the TAUs? These, of course, must be due to methodological differences in the scanner, noise quality. Yeah, so these obviously are not physiological, or certainly the vast majority of this variance is not physiological in nature. And if we did not do anything about it, we would never detect anything. And so it is essential to do this normalization. So this is what you get after you regress out age, gender, and ethnicity, and of course mean, because see, we're centered around 0. So that's a good thing. That's going to get these things normalized. But the other step that you always do, and just to maybe allay some of your fears, you might think, what is this magical thing, this inverse Gaussian normalization? It just magically makes everything Gaussian. And the technical name for this is a probability integral transform. It's just a way of you basically convert the data to ranks, and then you replace the ranks with what a normal variate would have for that ranked value. And so it seems magical. There's nothing wrong with it. In fact, we could do all of statistical analyses this way, but it's not perfectly efficient because we have to estimate the mean and the standard deviation to do this. So there's a slight loss of efficiency. But the most important thing this thing does is that it removes outliers. So if we have outliers that are way out here, those could give us real trouble because methods based on covariance are extra sensitive to outliers and can be really thrown off. So this is a good thing in general, and the point I wanted to make is that it's not just SOLAR. Basically, it's a standard pre-processing step in all of quantitative genetics. So it's I try to tell people that when I do this is a completely standard thing. There's nothing goofy about it. And in general, if the data are well behaved, it'll have no impact, and if you have outliers, it should make things better behaved. So anyway, this is crucial, standardizing and taking care of the nuisance variables by site, and then doing this inverse Gaussian normalization. It ensures univariate Gaussian normality. But something that is kind of a cute thing is that this does not guarantee that it's multivariate normality. The assumptions of this model is that there is multivariate normality. And my favorite example of how you can have univariate normality but not multivariate is here. So I simulated Gaussian variable 1, Gaussian variable 2, and with just a slight sleight of hand, I move things around. This data, this is the bivariate scatter plot for these two variables. marginally, if I project the data down on the x-axis, this is what it looks like. It's Gaussian. I could have shown you a QQ plot, it'd be perfectly straight. On the y-axis, projected on that axis, it's perfectly Gaussian. Is it bivariate Gaussian? Hell no. It's totally screwed up. That's any kind of test. Well, not that there are good tests for bivariate Gaussianity or even multivariate Gaussianity, but any sensible test would say that is not Gaussian because basically you have zero probability of getting anything in there. So, of course, you're not going to run into that in real data, but it's just a point that just because we have gotten rid of univariate outliers doesn't mean that there can't be bivariate or multivariate outliers. And then from Anita's plot, this is just one slide to just say that to remind us of the magic of meta-analysis. So just to stress this, these boxes here are representing the sample size. So they're just relative illustrations of, so this is a bigger sample, that's a smaller sample. And these lines here are the 95% confidence intervals. Well, the size of these diamonds are the 95% confidence intervals. So it's just sort of a visual thing to say, look how well we're doing. I mean, this is the variability in the study. We have incredibly more precision by doing a meta-analysis. We've gone from, if we were stuck with just a tau study, we'd hardly know at all where heritability is, and you see how much better we're doing. The other point here, too, was to compare the different types of meta-analyses based on just n, or standard error, or mega. And the interesting thing here is that this is being pulled a little bit more towards the standard error. So this is a relatively small study, but look at the standard errors. They're rather tight. So the method that's weighting the estimates based on the standard errors is a little bit more pulled towards that one, because it's basically saying, regardless of the sample size, that is a highly precise estimate of heritability. And we're going to sort of steer ourselves to that. Now, of course, not regardless of sample size. If this was a tiny sample size, if the noise of this was very large, this would still have wide samples. So this is relatively small, but it's a relatively precise estimate. And so it's not wrong that we're getting pulled this way, but they're just two different ways of getting a meta-analysis estimate, whether we just do it based on n, or whether we actually look at the length of these standard error bars. And that's what this method here is doing. And the mega-analysis is somewhere in between, which is probably I agree with Nada, it's probably a better way to go with it. It's just modeling all the data together. The other point I wanted to make from Nada's slides was she pointed out the cerebral spinal tract is got very low heritability, and she said this is because we have sort of methodological problems. It's rather unstable. Is that what you said? It's just kind of a quirky region to get. So that's just to remind us, the takeaway there is that, of course, what is heritability? Again, it's just this ratio. But, of course, there's a decomposition based on this. And so if you increase, all things equal, if you just increase the noise, so if you take something that has good heritability and then you just add white noise to it, just mess up each individual's measurement, well, of course, heritability is going to go down. Have you changed the biological constituent? Have you just take a good phenotype and you add noise to it? Have you changed the genetic control of that entity? If I take brain volume measurements and I just start adding white noise to it, have I made brain volume any less under genetic control? No. This is just intrinsic to what heritability is. Heritability is a fractional measurement. It's what percentage of the phenotypic variance is explained by genetic sources, and if this gets inflated, heritability is going to go down. So that's, I guess, just to the point there is that we're not saying that the cerebral spinal tract is not under genetic control or that it's special genetically. Rather, this is more a statement, I would say, that this is special from an MRI point of view. This is a difficult region to measure, and so that's why it's under relatively low genetic control. In fact, this is a point that Danny Weinberger made yesterday, which was he was making this comment about heritability of measures, of structural measures, and how far away they were from the anterior commissure, because of course if you're doing registration, things that are closer to the anterior commissure, or more practically, things that are closer to the center of the brain and the brain structures that vary less between individuals, subcortical structures, there's just less variability, and there's more variability farther away. And so again, that's not an intrinsic genetic thing, that's a methodological thing saying it is harder to register the cortex than it is the thalamus and the putamen. And so that means there's going to be more variability and hence harder to have high heritability at peripheral regions. So that's just something important. Don't just look at heritability measures and think genetics is, of course, it's a ratio. The sources, the explanation could be genetic or sort of methodological in nature. Okay, and then from Bennett's talk, he brought attention to the C effect. There isn't a lot of attention placed on the common environmental effects. So these are things that, for example, affect twins communally, not, or if there's a family. These are effects that might, if there's sort of, what would you say, pollution sources in a house or social economic status, something that is common to a set of individuals, but not necessarily genetic in nature. And so Bennett was pointing out that there are, definitely you can find some studies where there is. It's typically low, typically lower than our estimates of heritability for our brain imaging phenotypes, but it can be non-zero. You do see it. And I couldn't find the reference in short order. I can definitely dig it up. But from the twins literature, there is best practice that says that you basically always should fit the C effect. The issue is, if you omit the C effect, and that truly is there, that will inflate the heritability estimate. Because basically, if you have a true C effect, if there's something about households that make each other similar, well, and you omit that, that's going to start to look like genetic similarity, because that's the only other explanation you have why individuals would be similar in a household, if you don't have the C effect there explicitly. So it's something that it's always worth considering. Putting that, you always should have it in your model, just as kind of a safety. Even if it comes out near zero, you can sort of be safe in the knowledge that it's not, that any significance you are getting on H is due to an on-modeled C effect. And so, of course, common environmental effect or family effect in, household effect in family studies. Right, so that's all I wanted to say. I'm really happy that everyone has stayed here, especially the people who are not speakers in this symposium who stayed. You do outnumber us. And this is a project that, we're very grateful to Peter for leading the way. And I think this is a great tool. And basically there is a movement in genetics, traditional genetics, that there is a lot to be gained from studying families. If John Blangiero was here, he would have talked about the need with rare variants and how they're our best hope of finding very rare things when you have lots of related individuals. And it's very hard to find. If something's very, very rare, well, that means it's going to be very, very, very hard to find in any given number of individuals. But if you have people who are related, at least if something is rare, you have multiple chances of picking up a rare variant in a related set of individuals because they share genetic material. So there's a lot of interest in doing families and related individuals, and SOLAR is a tool that can handle that, both for getting at the heritability as a measure of interest or as a nuisance effect. So with that, I'll stop, and thank you for all sticking around. Hope to see you next year.
Can I ask you a question? Sorry.
Yeah.
But, I just thanks for reminding me about that statement that Danny made about, the Kinship stuff that's outside that gets farther away from the anterior commissure gets to be less and less genetically heritable.
Mm-hmm.
But if anything, our study shows that, corticospinal tract, which is sort of right there in between anterior commissure and posterior commissure, is going appears to be much less.
Well, he wasn't talking DTI.
It's because the ROI we're using is kind of lower down, and a lot of the brains actually have that more.
Okay.
That's why we're getting this methodological error, because some sites are able to capture the full region, some sites aren't. And it's also really noisy when you do DPI imaging. That's the part that really moves around.
Yeah, so I think the lesson there is that when you're comparing lots of things, you should always look at the total variance and just interpret the heritability relative to that.
Right, because if anything, we didn't see much, like in the proper tract, we didn't see much difference, so to speak, as a distance from anterior.
Well, so for the thing with the total, which is kind of tricky, is that when you do this variance normalization, when you do the inverse Gaussian, you basically, you don't put back in the phenotypic variance. So you basically standardize. So everything, when you start feeding it into SOLAR, is unit variance. So for the purposes of the analysis, because we are only concerned about h squared, we start with that all being equal. But with the mega-analysis, yeah, that's actually a good point. So basically you're pointing out that if you have differences in data quality between sites, you actually would expect heritability to systematically differ between sites. And that would be a shortcoming of the mega-analysis approach, which assumes that it's common, that's the same for all sites. Was that your point?
I was just asking, clarifying what you just said.
Yeah. Well, okay, so what it gets at, and we haven't talked about this, is in traditional meta-analysis, not imaging meta-analysis, just regular old meta-analysis, it is considered best practice to fit random effects models so that you allow for not just the variability reported in each study with their standard errors, but you allow for the fact that there might be systematic differences between studies. And the best practice at present is that you test for it, and then if it's present, you model it. And so what you are alluding to is that, in fact, yeah, it's reasonable to think that there might be differences in the true h squared that you observe, and that we should have some way of dealing with that.
Yeah, and then correct me if I'm wrong, but I think they actually tested for the difference, the significance of the difference of heritability among the tracks. And for the majority of the tracks, they observed there is no significant differences in heritability among the tracks. I think only for the spinal tract and perhaps for fornix, they observed that there are indeed significant differences in heritability among the sites. That was one of the arguments.
We have 5 sites.
Right, it's 5 sites. So, and this was one of the arguments we used to sort of, mark fornix and corticospinal tracts as area where the phenotypes may not be, reliable enough.
Because they're different.
Right, because they're different, because there are significant intersite differences between. And in terms of using mixed effect modeling, I mean, in many ways, I think it's desirable, but can you actually do it with just 5 sites? Because I understood that you need a lot more.
That's right. That's why we didn't consider it for this work. That's the problem. You now need that much more data to estimate the between.
Because, then the degrees of freedom becomes, the number of sites becomes the degree of freedom. And so when you have a degrees of freedom of 5, I think you subtract another 1 or 2 before you can actually calculate the numbers of degrees of freedom.
Yeah. It's not a linear model. I mean, it's another iterative thing, but it's it's a pain. And the other aspect is that I'm not even sure what the right thing is because h, of course, is bounded between 0 and 1, and random effects is always cast in terms of Gaussian random variation. So I'm not quite sure what would be the best thing. But it does. I mean, really, what sigma squared g, the genetic variance should be comparable. If FA means FA means FA means FA, that should be comparable between sites. And so you should be able to actually look at sigma squared g per site, and then you could compare. Yeah, well, you can make inference on that and see if that's very different. Yeah, sorry.
In SOLAR, we parameterize and analyze the model instead in terms of heritability and just estimate variances. You could test it directly there.
Okay.
Right. So I mean, we sort of don't do that. So instead, they actually use the inverse Gaussian normalization to make sure that they actually have the same variance per side.
But he's making a good point. So I mean, consider that the FA is bad because that too is bounded. So let's talk about bold. So let's say you're doing a study of bold fMRI, and some sites had 7T and some sites had 3T. So you might think, and then let's also make a big jump, which is maybe not correct, which is that the percent fold change, if you have a true physiological change, the same physiological change will give rise to the same percent fold change in both cases. But you know that the noise level is going to be different between those two sites. So you can have a case where in percent fold change units, that means the same thing between both sites. But because the one site with 3T is going to have more variability after the Gaussian transformation, that bold variance is going to be squashed down more relative to the 7T site. So that's the downside with the inverse Gaussian normalization. All you have to go by is the total variance you see. But if that total variance, that E, is systematically much greater at some sites than the other. Now, in general, I'm actually not that worried about that because what dominates is the between subject variability. And 7T versus 3T, that's going to be the measurement error on each subject. So I would actually be much more worried about a site that has, I mean, say they're all 3T, I'd be much worried about like a hospital site that has a clinical scanner that no MR physicist has looked at in a year. That would have much more variability that could have even like actually blow past inter-subject variability relative to 7T versus 3T on well-maintained scanners.
Right. And since in many ways it's basically like a variance analysis, then I think what's more important is to have consistent sort of consistency of measurements per family, so to speak, right? So if you have variability in, well, actually obviously stability across the entire phenotype, because if you don't have this stability per family, then you don't
Yeah.
have any.
Actually, no, and I talked about John, I talked about this with John. He said that actually that comes up in linkage studies, he's done variance components with linkage, where you actually allow different families to have different, yeah, so that's actually a standard thing from linkage, so it's possible. It actually may not be that hard to consider doing that. Yes?
Peter, when you were talking about the different types of data sets you could use, you mentioned family data sets. You also said that SOLAR can actually use unrelated. And then when Netta spoke, you know, there was only like 4 different examples of what small families to large families look like. And then, Tom, you reiterated again that right, heritability has to be based on some level of relatedness. That's how the matrix is made up, and that's how the thing actually
Yeah, so they could help you estimate the X beta part,
Right.
but then they can't help you with the heritability. And I think there'd be more of a case for them, because you can imagine, if you could do heritability with like 10, 20 subjects, and you have vanishingly small power with that. And if it could help to add in unrelateds, that would make sense, and I think there'd be cause to do that. But it doesn't. It doesn't. I've tried it, because the heritability is only coming from the contrast of the different degrees of relatedness, and piling on more unrelateds just doesn't help you. So if that did work, there'd be a case for piling on a bunch of unrelateds to nail down age, gender, and what have you. But
Greg, we can make another, but on the other hand, we can make a case if you have a very, very large unrelated sample, say 10,000 people, then, and you have GWAS for all of them, that's actually Meta is investigating this right now. You can actually build a pedigree out of them because
well,
everyone.
this is GCTA, right?
Right. Everyone is still related to each other. So you can actually build a pedigree matrix of kinship coefficients. They're going to be very low kinship coefficients. But you can actually run it and you can calculate the heritability. In fact, I mean, Andrew was taking it here. So people have done it on the ADNI cohort. And it appears that the people in the ADNI cohort, albeit they are unrelated to each other, they actually are related to each other at some sort of either 6 or 7 level. So you can actually calculate heritability from ADNI phenotype.
Have people done that? What sort of estimates do you get?
I'm not sure what the data really
Okay,
shows, but
okay.
there are a series of papers doing those kinds of calculations.
Okay,
And,
cool.
And I gather it's somewhat controversial in terms of the accuracy of the models. But it would certainly be convenient if you don't have twin sets for those of us doing case control designs to be able to have some heritability estimates.
Yeah.
Right. I think it's only controversial in as much as, ADNI cohort really has only 700 subjects. And most of the kinship coefficients are like at 0.001 level. So your measurement is highly, highly noisy. So it's kind of like mining the noise. But I mean, at 10,000 subjects, you definitely should have enough, big enough matrix where the noise can be rejected.
I'm not sure what the goodness of fit is, though, where you have both sources of information in those heritability estimates. I mean, if it's really accurate, you should see some comparable metrics coming out of twin studies and other kinship-type designs, and looking at these large.
Well, that's the question. That's why I asked about the estimates. Yeah, if they get, because presumably like whole brain volume should come in around 80% or 70% or something.
I mean, people.
It did?
I think that was the result from Andy, but the confidence intervals were really large.
And are they, so these very tiny estimates of relatedness, these coefficients, I guess, that populate the matrix in unrelated individuals, so is that based on alleles identity by state? And then you say, okay.
Oh, you just yeah, that's all it is. You just,
So basically it's allele sharing.
yeah,
And so they make some estimate of how related they are, and it's tiny.
Yeah.
Because,
on average, people are only distantly related.
But there was a study in Nature Neuroscience where they looked into the heritability of pi, wasn't it?
That was the first study that
Right.
used that method. Yeah,
They've actually
Fisher.
achieved a pretty nice heritability figure, and the confidence intervals were somewhat.
But they had 10,000 people, right?
They have more than
There's 100,000.
All right. Well, I think I will close this session and the post-conference conference.